In today's class, we'll be studying on plant-water relationships. In plant-water relations, we have the concept of imbibition, diffusion. We exactly will find out the answers what happens when you take a plant cell and put it in a beaker with normal distilled water, a cell which is immersed in a solution with higher concentration of sodium chloride. What happens when resins are put in water? Why do wooden doors swell during rainy season? As well as we'll also look at relations wherein how does water move into plant cells? When water moves into plant cells, what causes turgidity? How does a cell behave when it is put in an isotonic solution, in a hypotonic solution, as well as hypertonic solution? When you take the chapter of plant-water relations is predominantly determined by thermodynamics. The study of thermodynamics or principles of energy with relation to living cells is termed as bioenergetics. So, in bioenergetics, we have to be dealing with relation to any particular molecule, and the molecule which has got a charge, and by means of the presence of the charge as well as due to the presence of the molar nature, it would be having a little bit of energy. So, we would be studying how in a particular molecule potential energy is converted to kinetic energy, and when potential energy is converted to kinetic energy, what really happens. We take the first instance of imbibition of water. During imbibition, when we have a seed, the seed can adsorb because it is made up of hydrophilic colloids. Thereby, any water can be taken up by the seed coat, and the water which is in the form of a liquid can enter through the pores of the seed, especially the pores which are rich in cellulose, which are rich in pectin, and also the cytoplasmic proteins. So, the cellulose, pectin and the cytoplasmic proteins which are present on the seed coat can adsorb the liquid water, and thereby it develops a matrix potential. That is, it is capable of doing work. So, any liquid water which is present on the surface of the seed material, which is taken by the colloids, hydrophilic colloids. The hydrophilic colloids are present in the pores of the cell wall. They are rich in cellulose, pectin, cytoplasmic proteins, and thereby the matrix potential is increased. So, this is what which happens in imbibition. So, in imbibition, if we have an imbibant, that is the chemical which absorbs water is termed as imbibant. The volume of the imbibant increases. The volume of the proteins increases on the seed coats. This leads to germination of seeds. And during the germination of the seeds, the seed coat swells. Leading to breaking of the testa. So, in the process of imbibition, we have increase of the imbibement volume leading to all these changes. Normally during imbibition, what happens? The potential energy of the solute or the solvent is converted to kinetic energy. It has got the capacity to do work. So when it has got the capacity to do work, you call it as a potential. And the potential is attributed to the chemical. Hence you call it as a chemical potential. Or you could also call it as the potential or the capacity to do work in terms of the water. So you could also call it as a water potential. So any normal biological compound from potential energy is converted to kinetic energy. And when it gets converted, the heat of energy is generated. The heat which is generated leads to the breakdown of the testa. Similarly, when you take imbibition, another example of imbibition is how roots absorb water. Roots also absorb water. This is a second example. Roots absorb water by the process of imbibition. Seed coats swell, especially when you take legumes. So imbibition is more in the case of legumes when compared to the cereals. So proteinaceous substances show more imbibition when compared to carbohydrates, when compared to cellulose. Proteins have greater value of imbibition. The entry of water into ovules prior to the formation of seeds is also an example of imbibition. Wooden doors swell in winter, swell during winter as well as in rainy season due to the process of imbibition. So here when you take the proteins as indicated show higher imbibition when compared to agar, when compared to starch, as well as when compared to cellulose. So highest imbibition is seen in the case of proteins. It is less in the case of agar and starch, and it is less in the case of cellulose. So here imbibition is due to matrix potential. And in the case of the solvent, because potential energy is converted to kinetic energy, the value of the matrix potential is always negative. We go to the second concept termed as diffusion. During diffusion, the water from the soil, how does it enter into plant parts? How does water move from one cell to another cell, especially from a higher concentration to a lower concentration, is termed as diffusion. So during diffusion, if you have any molecule, the molecule is normally in a free energy state. The molecule has got more of movement. So when it passes through a semi-permeable membrane, this is a semi-permeable membrane. So from free energy, it can primarily undergo the process of diffusion. During diffusion, there is a directional movement. there is a directional movement. So here what happens is, primarily the solvent, that is the water, moves from an area of free energy into a solute with a solvent. Thereby the free energy is reduced, whereas here the free energy is very, very high. It is capable of more of kinetic energy. There is more of collisions. It has got higher potential. The potential is attributed to water. It is also termed as chemical potential. So from a region of higher water potential or higher chemical potential or higher free energy, when water molecules move across a semi-permeable membrane and go to a place where there is lower free energy and again lower water or chemical potential, this property is termed as diffusion. Now when you take diffusion, during diffusion there is, the common example is exchange of carbon dioxide and oxygen. Exchange of carbon dioxide and oxygen, as well as the movement of water in interstellar spaces, in the leaves. And again we also have passive salt absorption. Passive salt absorption, exchange of carbon dioxide and oxygen, as well as movement of water through the interstellar spaces. All these three are examples of diffusion. Diffusion is a passive phenomenon, but again when we come towards osmosis, in osmosis we have the concept of membranes. Now membranes in biological cells can be of three different types. They could be permeable. A is they are permeable. So permeable means they allow solute and solvent to pass through them. A common example of a permeable membrane is a cell wall. They allow the solute, and both the solute and the solvent can pass through a permeable membrane. Second we have is impermeable membrane. An impermeable membrane will not allow any solute or solvent to pass through it. It is primarily rich in lignin, suberin and cutin. Cutin is present on the surface of the epidermis. Suberin is present in thick walls in trees. Lignin is a brown compound which prevents any particular pathogen from attacking the plant. So all three of them, lignin, suberin and cutin, are examples of impermeable membranes. We come to the third category, which are termed as semi-permeable membranes. Now semi-permeable membranes are also termed as selectively permeable membranes, or else they are also termed as differentially permeable. They are differentially permeable. They allow certain solutes, but always they allow all the solvents. So for all the solvents it is yes, for solutes it is no. They do not allow solutes. There are exceptions to this, but mostly they allow all the solvents. So any membrane which allows all the solvents to move through this is termed as selectively permeable membrane. when you take the plant cell, in the plant cell we have the cell wall, we have the plasma membrane, and we have the tonoplast. When you take the plasma membrane and the tonoplast, they are examples of selectively permeable membranes. We come to the process of osmosis. Whenever there is osmosis, osmosis is a directional movement of water molecules. Whenever we have water molecules move in a particular direction. And osmosis, to begin with, is passive. It does not require any ATP. It occurs from any particular region of high free energy, that is where the entropy is available in terms of the molecule, where the molecule has more kinetic energy. So, the kinetic energy is high. From a region of high kinetic energy, thereby there is more of collisions. So, because it has got more of energy by view of its chemical nature, you can also call it as it moves from a region of high chemical potential. From high chemical potential, or you can say even from high water potential. The capacity of free water to move is termed as water potential. Now, when you take pure water, in pure water the water potential is designated as 0. In pure water, the water potential is given as 0. Now, when this sort of energy of any particular molecule, preferably water, when it moves through a semi-permeable membrane, or a selectively permeable membrane to another region of lower free energy. From higher free energy, it goes to a lower free energy. And where there is low free energy, there is more of concentration. Why do you call it as concentration? Because there is presence of solute in a solvent. More of solutes are present. So, the solute dissolves in the solvent. Thereby the water loses its energy, free energy. And here this process is predominantly termed as endoosmosis. So, during endoosmosis, it is directional, passive, higher free energy to lower free energy across a semi-permeable membrane till equilibrium is achieved. And normally it occurs from higher water potential, chemical potential and kinetic energy towards any other place where there is more of concentration of the sort of solutes. In osmosis, we are looking at what happens when a biological cell is placed in distilled water. We have a biological cell, biological plant cell. If the biological plant cell is placed in a beaker which contains distilled water, the biological plant cell is rich in cell sap. It has high osmotic pressure. Osmotic pressure is because of the cell sap, and because of osmotic pressure it exerts a pressure which is also termed as turgor pressure. Because of the turgor pressure, the cell is turgid, and the parallel back pressure which is exerted by the wall is termed as wall pressure. So, in a biological cell sap, it has got high osmotic pressure, turgor pressure and wall pressure. Soluble solutes that is solutes which dissolve, can enter into the plant cell, by means through the distilled water, and due to this the process is termed as endoosmosis. It is termed as endoosmosis. So it occurs through the semi-permeable membrane. The semi-permeable membrane is the plasma membrane. Thus, normally when you take a biological cell and place it in distilled water, it exhibits endoosmosis. The reason is because of A, higher cell sap, high osmotic pressure, turgor pressure, as well as wall pressure, and entry of soluble solutes. From the distilled water, it enters into the cell sap. What happens when the cell is turgid? When the cell is turgid, a cell is compact and you call it as turgid. In turgid condition, the hydrostatic pressure the water pressure or the hydrostatic pressure is exerted by the cell sap. So the cell sap exerts a pressure and the pressure is termed as a hydrostatic pressure. So when there is a hydrostatic pressure, further entry of water from the outside is prevented. Because the cell is already turgid. Now, when it's turgid, the cell sap has got solutes. The solutes have a potential, that is, they have their own energy, and the solute in a solute potential is always negative, is given by a negative symbol. Parallelly, we also have the pressure. The pressure also, it results in the formation of a pressure potential. The pressure potential is due to the presence of the cell wall, and even this one is negative or negative, designated like this. So when you take a turgid cell and you place it in water, water is prevented from going in primarily due to hydrostatic pressure. Now in a turgid cell, both the wall pressure and the turgor pressure become equal and opposite. Turgor pressure and wall pressure become equal and opposite. Because water cannot enter, the water potential is equal to 0, is given as 0. So that here when you take water potential, it is 0, and when you add any particular solute, the value of water potential becomes negative. The older concept was you attribute to water potential as diffusion pressure deficit. DPD was an older term which was used for water potential, and the value of DPD in any equation is given as a positive value. It should be understood DPD has got a positive value, whereas water potential is 0, and normally water potential is given as a negative value. DPD has got a positive value, water potential has got a negative value, especially when cells are turgid. In turgid cells, what happens when cells are flaccid? When you say flaccid, it means cells shrink or the cell will collapse or it loses turgidity. When you take a flaccid cell, in a flaccid cell, the turgor pressure is equal to 0. It does not have turgidity. And turgor pressure is 0, especially when there is water scarcity. When you take desert plants or when plants are under stress. So thereby the osmotic pressure or solute potential is equal to the water potential. So when a cell is placed in water, water enters due to higher dPd of the cell sap. So you have two situations. One is what happens when cells are flaccid, how it occurs under water what happens with the turgor pressure, especially when there is water scarcity, what is the water potential, how is the value of dPd, and why does water enter? The water enters because there is high dPd of the cell sap. Now when you take a grape resin, a grape resin is dried grapes, and you put this sort of a resin in water. When grape resin is put in water, initially the cells are flaccid. The flaccid cell regains shape. It can regain the shape because primarily on the outside you have hypotonic water solution. The water solution on the outside is hypotonic, lesser concentration. So because it has got lesser concentration, it will have low osmotic pressure. Now when you take the diffusion pressure deficit, dPd, the diffusion pressure deficit is equal to osmotic pressure minus turgor pressure. So there, because of this, the cell has high diffusion pressure deficit. It is rich in sucrose, it's got high diffusion pressure deficit. Thereby the water moves from the outside to the inside. There is influx of water. There is influx of water from the outside solution into the sort of grapes, and this leads to swelling of the sort of grapes. Grape resins, when put in water, primarily they swell, and this is due to endoosmosis. It is due to endoosmosis. Next, under osmosis, we come into the concept of the water potential. The term water potential has been studied by Slater and Taylor. Normally, water potential is designated by the Greek word with a symbolic representation of a omega with a W. So, normally water potential, during water potential, the movement of the water is from higher water potential to lower water potential. That is, from a value of 2 bars to a value of minus 3 bars. Now imagine minus 2 indicates higher water potential and lower water potential is indicated by minus 3 bars. Again, water potential is given as a addition of solute potential S plus also the sort of a pressure potential which is present in any particular cell, and the value of water potential always is equivalent to diffusion pressure deficit. Water potential is given as a negative value, whereas diffusion pressure deficit is given as a positive value. Water potential for a cell is a combination of the solute potential as well as the pressure potential, and it primarily gives the direction in which the water, this solvent, moves within a particular cell. What happens in a plasmolysed cell? Supposing a cell has shrunk, you have a cell, the protoplasm shrinks, this is the cell wall, and we have a plant cell. The protoplasm in turn, it shrinks, goes on the inside from the cell wall, and this process of shrinking is termed as exoosmosis. And it when it shows exoosmosis when it is placed in a hypertonic solution. When the solution is hypertonic. Like if you take a cell and put it in 0.2 molar sodium chloride. When you put it in sodium chloride, the cell normally shrinks. So when it shrinks, the property of the cell, of a plasmodized cell, is the cell becomes flaccid. The cell becomes flaccid in a hypertonic solution. In a plasmolysed cell, the Turger pressure is 0. Diffusion pressure deficit equals osmotic pressure. Now, diffusion pressure deficit is equal to, is given as osmotic pressure minus Turger pressure. Turger pressure is also always termed as 0. So, normally what happens is, when you take cells and you put them in saline water, when you put them in saline water, it shows plasmolysis. Similar is the case when you take leaf cells of Tradescantia. Lower epidermis leaf cells of Tradescantia, when you place them in a medium with, say, 10% of sodium chloride, when you put them in a salt solution, the leaves will collapse. So here what normally happens is, from a hypotonic solution, there is the movement of the sort of solutes to a hypertonic solution. From within the cell, it goes to a stronger solution which is present on the outside, thereby it gets plasmalized. Now, there are two very important concepts which we need to understand. In a plasmalized cell, normally is asked in K-set. In a plasmalized cell, the space between the cell wall, you have the cell wall, then you have the plasma membrane. The space which is present between the cell wall and the plasma membrane, this space is rich in hypertonic solution. That is, the concentration of the solution is very, very high here, whereas the space between the nucleus and the plasma membrane, this is normally occupied by an isotonic solution. An isotonic solution is always in equilibrium, and there is no increase, there is no cell increase or cell decrease, whereas when you hypertonic solution normally resides between the plasma membrane and the cell wall. Now we are coming to osmosis. Osmosis is a method of diffusion. It relates to movement of water across a semi-permeable membrane. It allows the movement of the solutes freely, and it is an example of directional diffusion. How does osmosis differ from diffusion? It is more directional. The movement of the solutes is always from a region of high to lower concentration. And always the forces, especially with relation to potentials, are equal and they are opposite. So the basic attributes of osmosis are, the properties are diffusion of water. It primarily relates to diffusion of water.